and we are on. So I believe this is chapter 9 in your textbook. I have an older edition of the textbook. They're all about the same. The editions, they just add new pictures and a few new words if they discover something new in AP, which is rare these days, but it happens, especially at the microscopic level. So the reason I pause sometimes is because I'm admitting students as they drop into the waiting room. So let's go ahead and get started with the muscular system. The first thing I'm going to do is just call up a whiteboard. I can't write every single thing out. It just we wouldn't get anywhere. I wouldn't get to the material that I need to get to. So I jotted down a list of things I want to cover, but I am going to use my whiteboard to just give you highlights, and I will talk, don't worry. We did this when we studied tissues, 3 types of muscular tissue. So I'm just going to quickly go through their characteristics again. First one is skeletal muscle. And the reason it gets its name, skeletal muscle, because this is the stuff that's attached to bones. It's the specific muscle tissue type. Yeah, Patrice, I got your message. Sure, no problem. So this stuff is attached to bones. Just to reiterate the characteristics of skeletal muscle, these have the longest fibers. Before I go any further, when you hear me say the word fiber, or you read the word fiber, it's the same thing as a single cell, a muscle cell, especially when we talk about skeletal muscle. So muscle fiber means the same thing as a muscle cell in this case. The reason is because they're long and thin. And skeletal muscle fibers tend to be longer than the other 2 types. They also have striations. Those stripes, when we look at them under a microscope, they are voluntarily controlled. That means we have conscious control over the contractions. The cells are multinucleate. So the cells have many nuclei. And when we get into this unit, I will tell you how that's derived, where they get their multinucleation from. And finally, this is important, and I didn't say this in our tissue section. Each cell must be neurally stimulated in order to contract. So in order for a single cell to contract, each cell must be neurally stimulated. So that's going to be, you'll see that's unique compared to the other 2 types. So I've gone through skeletal muscles characteristics as a review. So I'm just, if you came in late and you want me to allow your recording, I'm recording this. Just send me a chat, type me a chat, and I'll enable your recording, no problem. So the second type of muscle tissue, as you know, is cardiac muscle. To reiterate some of its characteristics, it's only found in the heart. Obviously, it's not attached to bones. It is striated. It has those stripes under microscopy. It is involuntarily controlled. We don't have conscious control over it. The individual cells or fibers have 1 or 2 nuclei per cell. So it can be multinucleate, but nothing like skeletal muscle. Each individual fiber has many nuclei in skeletal muscle. Cardiac, 1 to 2. And cardiac muscle does not need to be neurally stimulated in order to contract. When you take A&P2, you'll learn why. But an actual nerve cell does not need to innervate cardiac muscle fibers in order for them to contract. Finally, the third type, smooth muscle. This is the stuff that we find lining our visceral organ walls as well as our vessel walls. It is involuntarily controlled. We don't have conscious control over it, just like cardiac muscle. There are no striations. That's where it gets its name. So this one gets its name based on its location, this one because it's attached to our bones, and this one because it doesn't have striations when we look at it under a microscope. Smooth muscle fibers or muscle cells have one nucleus per cell. They are not multinucleate. And this one needs to be neurally stimulated in order to contract, just like skeletal muscle. But the neural impulse, electrical impulse, can be transmitted from cell to cell. I know we haven't done the nervous system yet, so this may or may not make a whole lot of sense to you. But in a skeletal muscle fiber, if we have a muscle, a big muscle that's made up of hundreds or thousands of individual fibers, each individual fiber of skeletal muscle must have its own nerve ending in order to contract. Physically have its own nerve branch or ending coming in to stimulate it. That's for skeletal muscle. Smooth muscle, on the other hand, if we have a whole group of smooth muscle tissue, those cells kind of look like this. They're not as regularly arranged. They're sort of arranged in sheets. So I'm drawing smooth muscle cells now. They do need some sort of physical neural stimulation, just like skeletal muscles. However, the cell can transmit that electricity from cell to cell. So each individual cell does not need its own nerve like skeletal muscle. So we can get the whole sheet of muscle to contract by just stimulating it in much fewer places. And then the electricity can actually be transmitted from cell to cell. All right, let's move on to this. I will expect you to know general characteristics of muscles. And I'm going to quickly just highlight those. This is easy to understand. It's not complicated business. I'll go in the order of your book. These are words. It's just 4 words that end in ity. And the first one is excitability. Excitability just means that muscle cells, that characteristic of the word excitability just means that muscle cells can receive and respond to some sort of stimulus. So when, for example, a nerve cell releases its chemicals onto a muscle cell, something happens. It excites it. It causes a change in the membrane. That's excitability. Next characteristic is contractility. That just means that the cells shorten. Usually upon excitability, those cells can get shorter. And in the shortening process, they produce some sort of force, work, or tension. By the way, contractility is a unique characteristic to muscle cells in the human body. Next characteristic is extensibility. That just means that muscle cells can extend or stretch. They have the ability to get longer when forces are acted upon them. And finally, elasticity. That just means that the cells recoil after they do change shape, after they either compress, shorten, and after they stretch or extend. So we've got excitability, contractility, which is unique, extensibility, and elasticity. Next, I'm just going to mention the functions of muscles. and I'm going to go in the order of your book, I believe. Excuse me. Muscles produce movement in the body, both what you and I think of as movement when we move our arms, legs, head, whatever, skeletal muscle movement, as well as smooth muscle movement. Our vessels dilate and constrict. Our digestive organs churn and the walls contract and move. So movement is a big one. Maintenance of posture. So the whole reason you can, I assume you're sitting in a chair right now, so am I. The whole reason that we can sit up is the tonicity of our muscles, the contractile state, and then pulling on bones to allow us to sit up straight or stand up straight. They also stabilize joints. One of the last things I said in the joint lecture is that the strength of a particular joint, specifically synovial joints, are largely influenced by the muscle tone around those joints. They generate heat, so they do help our bodies regulate body temperature. They protect. Protection is one. Protection in the form of organs underneath the muscular tissue, all around our abdomen, our abdominal cavity. It is largely muscles that protect our abdominal organs because very little to no bone structure except in the back of the vertebral column. Protection, they also regulate muscles, regulate valves. So valves are just opening and closure, entrances and exits, pathways through body systems, and those entrances and exits are typically regulated by muscles. And finally, muscles can store nutrients, particularly glycogen, which is a stored form of glucose or sugar. All right, that takes care of general functions of the muscular system or muscles in general. Now I'm going to get on to, I'm just looking at my list that I jotted down that I want to cover. Muscle structure is the next thing in your textbook, so I'm going to cover that. The structure of skeletal muscles. From this point forward, we're going to largely just be talking about skeletal muscle. That takes up the bulk of the chapter in your book. So until we get to smooth muscles, sorry about the writing. When I say the word muscle, pretty much from this point forward, I'm going to be talking about skeletal muscles, what you and I think of when we hear the word muscle. So I'm going to go over the macro structure and then the microstructure. So the macro anatomy, what we can see with our naked eye, is what I'm going to talk about first. So there is typically a rich blood supply to these muscles. There's usually one major artery and nerve supplying skeletal muscles. I'm talking about an entire muscle now, not just an individual cell. And then there's usually many veins that drain that muscle. So what that means is this: if we have an entire muscle that's made up of thousands of individual fibers, there's one major nerve coming into this muscle. And then, yes, it branches to innervate each individual fiber. There might, like I'm drawing as such, and there's usually also one major artery that supplies this muscle, and it also branches, and it doesn't necessarily go the branches to each individual fiber, and there's tinier branches and capillary networks that actually infiltrate each individual fiber. The point being, one major nerve, one major artery, and then multiple veins usually drain this thing, take deoxygenated blood away from it. Also, we're going to look at the macroanatomy of this business and how the fibers are arranged. That means if we looked at a muscle, dissected it, we could see this stuff pretty much with the naked eye. Okay, first I'm going to go over the connective tissue sheaths. That just means coverings. So there's something called endomysium. By the way, myo means muscle. So any word that has myo in it, and this one has a derivation of myo in it, that just means muscle. So this means technically this means within muscle. This is a connective tissue sheath, endomysium. It's thin and delicate, and it wraps or surrounds each individual fiber. Okay? And then I need to call up a new board, so I'm running out of room. There's 3 of them. Then perimysium. Let me write this out and then I'll go to my new board. So we have endomysium, a thin, delicate connective tissue wrapping each individual fiber. Perimysium is a bit coarser. So I'm going to write coarser CT that wraps several muscle fibers into bundles. And those bundles have a name. And I'll put that in parentheses. Those bundles are called fascicles, F-A-S-C-I-C-L-E-S. So fascicle is just another name for these bundles that are created when multiple fibers are wrapped in perimysium. And finally, number 3, epimysium is the thickest, coarsest connective tissue that wraps an entire muscle. So epimysium actually wraps several fascicles together to make up an entire muscle. So really quickly, if we have an individual muscle fiber, it's long and thin, this is an individual cell, and we put a thin, delicate connective tissue around it on the outside covering. Now if I take several of those wrapped fibers, and I'm not going to put the blue around each one, it would just take too long, and I wrap those together in a thicker connective tissue called perimysium. The green is the actual perimysium. This bundle that I've created is a fascicle. And now if I take, I'm going to do this quickly. If I take several fascicles that are wrapped in perimysium, and I wrap those together in a really thick, coarse connective tissue, I have formed an entire muscle, and this is the epimysium. I've cut off the ends of this wrap of this tissue to form the entire muscle. Technically, especially the epimysium, it extends off the ends of the muscle like this to help form a tendon to attach it to a bone. So that's the macro anatomy of how a muscle is arranged. Next, we need to look at the micro anatomy. Before I do that, I am going to talk about muscle attachments because your book does it. I usually do it later in my lecture, but I'm trying to do things in the order of your textbook just to help you stay organized. So I'm going to quickly write attachments. And first, let's just define this. Origin. This will be helpful later in the muscle unit for you. The words origin and insertion refer to the ends of a skeletal muscle, the points of attachment at opposite ends of a skeletal muscle, at different ends. And the origin end of a skeletal muscle is typically the end of attachment that when the muscle contracts, there is no movement. So I'm going to write no movement during contraction. I'll give you some examples. The insertion end is oftentimes the opposite end of a muscle, but not always. It's just a different end of attachment. So I'm just going to write end of attachment where there is movement during contraction. So really quickly, the biceps muscle right here in your upper arm, the points of attachment are here, actually at the forearm, and the other end of attachment is up here on the back of our shoulder. There's multiple attachments up here. When this muscle contracts, my forearm obviously flexes like this, but the points of attachment are at the shoulder and here. When this bicep muscle contracts and shortens, the movement end is here. So we know that this is the insertion. Up here at my shoulder, no movement. That's the origin end. That's what we mean by those points of attachment, origin, and insertion. Let me go back to a whiteboard. Also, I'm still talking about attachments. Tendons, these are indirect attachments now, what are called indirect. An indirect attachment example would be a tendon, which is connective tissue that is in a rope-like shape that connects muscle to bone, skeletal muscle to bone. And oftentimes, the bulk of a tendon is the actual epimysium that comes off the end, like I said earlier. So if we have a muscle, an entire muscle that's wrapped in that epimysium, as that epimysium comes off the ends of that muscle, it's sort of like if you take a roast or a piece of meat and you lay plastic wrap on your countertop and the plastic wrap is typically longer or wider than that piece of meat. And you throw the meat on the plastic wrap and then you roll it up. The ends of that plastic wrap that come off the ends of that piece of meat would simulate, that's an analogy to the tendon then that attaches to a bone like this. So tendons are forms of indirect attachment. So is something called an aponeurosis. That's aponeurosis. Aponeurosis. This is just a flat tendon, basically. A flat sheet-like tendon. It's not rope-shaped. It's flat sheet-like tendon that it also attaches skeletal muscle to bone. And we have an aponeurosis coming off of our flat muscles. So think of your abdominal muscles that cover the anterior portion of your abdominal cavity, your abs, that group of muscles is a flat sheet-like group of muscles. And here's an anterior view of that group of muscles. There's a connective tissue covering over all of these that then runs off. It's a flat sheet-like tendon that runs off the edges and just attaches to a bone. In this case, it would be the pubic bone in front of the pelvis, and up here on the sternum and the lower ribs, that flat sheet-like tendon would attach up here. A direct attachment is typically referred to when the actual muscle itself, without a tendon or aponeuroses, attaches to a bone directly. All right, now I'm going to examine microscopic muscle anatomy, and that's going to take up the bulk of the rest of our lecture, because we need to build a word bank, a bank of terms that's going to help you understand the physiology later. So tomorrow I'll begin to do a lecture on how a muscle actually contracts. Today we're focused mostly on anatomical structures that's going to help you understand that physiology. So let's just start building a word bank here. Sarcolemma. Remember I said the prefix myo means muscle. Also this word sarco, that also means muscle. So whenever you see sarco, think of the word muscle. Sarcolemma is just the plasma membrane of a muscle fiber cell. That's what it is. So the endomysium, that thin connective tissue covering that wraps an individual muscle fiber, would be on the outside of the fiber sarcolemma, would be on top of that. Next term, sarcoplasm. This is just refers to the cytoplasm of a muscle cell. Rather than using the word cytoplasm, we use sarcoplasm because it's more specific. So there's all sorts of things going on inside of the sarcoplasm. I'm just going to write cytoplasm of a muscle cell. And we've got all sorts of things inside of there. Contains myoglobin inside of the sarcoplasm. Myoglobin is a protein-based molecule that binds or stores oxygen. Just like hemoglobin binds oxygen in the bloodstream, we use the word myoglobin. Myo means muscle. It's a very similar structure to hemoglobin that binds and stores oxygen inside of a muscle cell. There's also granules storing glycogen. Glycogen is a stored form of glucose or sugar. Inside of the sarcoplasm of the muscle cell, we also have glycosomes. The glycosome is just a term for the actual structure that stores the glycogen. Now we're going to look deeper inside of the sarcoplasm. So we're going deeper and deeper microscopically inside of an individual muscle fiber. A myofibril, I'm going to make it plural because there's many of them inside of a muscle fiber. These are, sorry, I was trying to write a P, specialized organelles. And they contain protein myofilaments This is just language. I know if you've never heard this language before, it might be a bit confusing. It's not complicated. It's just learning new words right now. So myofibril is just the name of a specialized organelle that contains protein myofilaments. Myofibrils are just long cylinders that contain the rod-like myofilaments. I'll draw a picture at the end of this. When we look at a muscle fiber, an individual muscle fiber cell, it's just like the muscle was arranged macroscopically, where we have long cylinders grouped, packaged together to form an entire muscle. Guess what? That's also what the inside of an individual cell looks like. It just looks like a bundle of tubes wrapped around a bundle of tubes wrapped around a bundle of tubes. These myofibrils take up the bulk of the volume of the inside of the cell. Now, we just stated that myofibrils contain myofilaments. What does that mean? What are those? These are the actual contractile structures inside of a muscle fiber. So these are small, sorry, I'm trying to spell two words at once, doesn't work too well. Rod-like protein structures inside of the myofibrils, and there are three specific myofilaments. Myosin, which we refer to as the thick filament, just because it's thicker in size. They're all protein-based, so they're all like chains of amino acids, by the way. The second myofilament is actin. Actin is a very common myofilament that occurs in many cells, not just muscle cells, to help with structure, but particularly right now we're concerned about muscle fiber cells. Actin is referred to as the thin filament because it's a peptide chain that is very thin relative to myosin. And finally, the third one is the elastin filament, which we also call titin, T-I-T-I-N. And this functions as a recoil for elasticity. So when we stretch a muscle, it's the elastin or titin myofilaments that help recoil that cell back to its original shape. Let's keep building up our word bank here. This will be really helpful for you for the rest of this muscle business. The word or term sarcomere refers to one contractile, that's a U, unit of myofilaments. Within a fiber. Once you look at pictures in your textbook, as well as I'm going to draw pictures, these words will start to make sense, to take on some sort of meaning. I'm going to say I didn't have room on my previous board, on this board, to say everything I wanted to about myosin and everything I wanted to about actin. So I'm just going to say a few more things about them on this board. So myosin, we know that's the thick filament. Here's some more detail about myosin. It's a thick, long polypeptide chain. That just means a group of amino acids. It's protein-based is what that means. Each individual myosin has a globular head on it. and a long tail. So it looks like this. I tell students, it kind of looks like a golf club. There's a large head on an end of a long stick or a long tail. So that's kind of what the myosin myofilament looks like. And it's on this head, I'm using a different color now, it's on this myosin head that it has a binding site to an actin filament. So I'm going to draw an actin filament up here, which just looks like a chain of long beads. And there's an active site on actin and a binding site on myosin that allows these two myofilaments to connect or attach to each other right here. So I will say actin is just, we know that it's the thin filament. It's a long bead of amino acids. So it's a polypeptide chain. They call it a globular chain. That's where it gets its bead-like or necklace-like arrangement or description. And these have active sites for myosin, the thick filament, to bind to. Okay. Tropomyosin, I know this is getting, it does get sort of head spinning with all of these new terms. Tropomyosin is also a polypeptide strand. So they all have a similar structure. They're all made up of amino acids and they're polypeptide in some nature. This is a polypeptide strand that covers and blocks active sites on actin, so that when a muscle is relaxed, myosin cannot bind to it. So I'm just going to use green. Tropomyosin sort of wraps around the actin molecule like this, like a grapevine, and it blocks the active sites on actin right here so that myosin can't bind to it. What we're going to find is when a muscle contracts, that tropomyosin gets lifted off of actin so that myosin can bind to actin. A few more words, and then I'm going to try to draw a picture to put this all together, okay? So we've got tropomyosin. The next, I have to go to a new board now. Troponin is a small sort of 3 polypeptide complex. So it's almost like a small block of amino acids. I'm going to write small polypeptide block. That binds, well, let me say this first. First, it's attached to tropomyosin. And it binds with calcium ions. That's the important part. Sarcoplasmic reticulum. That's supposed to be an R. S-A-R-C-O. It's like endoplasmic reticulum. Sarcoplasmic reticulum is an organelle inside of the sarcoplasm of a cell, of a fiber, stores calcium ions and associates with T tubules. Now I'll define what a T tubule is. Well, before I do that, this word, you're going to hear it and read it. Terminal cisternae are just bulging sacs. They're part of the sarcoplasmic reticulum. They're bulging sacs of, I'm just going to put the SR, the sarcoplasmic reticulum, that store the bulk of calcium. And they're the part that are associated with T tubules. So what's that? A T tubule is also it's considered an organelle, but really it's just a pathway. It's an invagination of the sarcolemma. That transmits electrical impulses from the sarcolemma, which is the plasma membrane, to the inside of the cell. So I'm just going to put to interior of the cell or fiber. These definitions are in your book, but it helps if you hear me say them and you watch me write the simplified version of what they are. Now I'm going to try to put this stuff together for you pictorially before we call it a day. So here's how an individual muscle, muscle cell is arranged. First, I'm going to quickly go over how myofilament. I'm going to draw two pictures before we leave. Myofilament arrangement is very specific, neat and orderly within a skeletal muscle cell. And it looks something like this. Most books draw it, some sort of version of this. So I'm going to draw a thick vertical line in the middle of this diagram and call this the M line. It gets its name M line because it's what myosin is anchored to. And then the thick filaments, the myosin thick filaments, sort of come off of the M line in a horizontal fashion like this. And they have globular heads on them, so I'm just going to remind you. So these are thick filaments, myosin. Myosin molecules. They're clustered together in groups. This is a simplified diagram, schematic diagram of sort of how the myofilaments are arranged deep within a cell. So this is magnifying a muscle fiber cell a gazillion times. We can't even see this much detail with the microscopes that we have in our own labs. So super powerful. This is the M line. These are thick filaments or myosin molecules, okay? Now thin filaments are arranged like this. The thin filaments are these horizontal black lines I'm drawing right now. So these would be actin molecules. Okay. I just left this one off because of my red arrow. I didn't want you to think I was pointing to the black line. The red arrow is pointing to the red. So this is an actin molecule. The red horizontals are myosin molecules. They're anchored to the M line. And myofilament arrangement within a muscle fiber cell is very regularly arranged, just like this. And it repeats, it keeps repeating hundreds of thousands of times all the way down an entire muscle. This is within one single muscle cell, muscle fiber. And then this arrangement just keeps repeating itself over and over again. Like this. And it would go on this way as well. I'm quickly drawing these not as neat, just to show you that it repeats over and over and over again, both in this direction and this direction. What I have drawn for you in the center of the page is one sarcomere, one contractile unit of myofilament. So I'm going to use a different color, from this Z band, and it gets its name because of its zigzaggy, to this Z band is one sarcomere. It's called the Z disc, actually. And one sarcomere, a sarcomere is one contractile unit of myofilaments. Now, it is this specific arrangement of myofilaments, this very regular arrangement through a muscle cell, that gives muscle fibers, skeletal muscle fibers, their banding pattern, their striations. So if ever asked, how do skeletal muscles, and cardiac muscles for that matter, but how do skeletal muscles get their banding patterns, their striations? It's from the regular arrangement of these myofilaments. That's what we see when we see stripes under a microscope in a muscle cell. Okay, I'm going to say a few more things about this banding pattern. We've defined the M line. This technically is called the Z disc. And you can see why. And it anchors the actin molecules. And just so you know, the ends of these myosin molecules are attached to a coil-like structure. That's that elastic filament called titin or elastin. That helps enable this cell to recoil back into shape after it should be stretched. From here, I'm going to use a different color because it's just going to get confusing if I use black. My eraser's not working. Oh, looks like I lost functionality of my pen, so I have to plug it back into my iPad. From the ends of actin molecules where there's no space, meaning right on either side of the M line, there's no actin molecules. That white space surrounding the M line is called, there's banding patterns. That's called the H zone, where there's no actin. The Z disc is where there's zigzag lines are. The I band is from, I wish this was working, the I band is from the end of this myosin to here. I'll review this tomorrow. My pen's running out on me today. So I'm going to plug it in for another 60 seconds while I talk, and then hopefully I can draw one more picture for you. It supposedly charges in a minute, but I found that that's not really true. You know what? Because I've run out of juice on my pen and I can't draw pictures anymore, I'm going to call it a day, and I'll pick up here tomorrow with tomorrow's lecture. So let me just go back to sharing my screen. And if you have any questions for me at all, either about muscle business or the bone quiz that you're going to take today, feel free to ask. I'm just going to cut my recording.